MIN is smart molecule of natural genetic resources. The focus of the research, we have three kinds of interests. One is nutritional genomic, is nutrition based on gene of human disease. The second one is pharmacogenomic. If in this group, explore the drug for management of the abnormality gene of human disease. The last one is gene mapping or fingerprinting, not only in human but also in animal and plant. We have now preparing the gene bank special for genetic variability in Indonesia. My research have a lot of publication and also join with some university in international collaboration with the Kassel University, Niigata University in Japan, and also Tokyo University of Agriculture in Tokyo. Our product research based on the goat millet from goat Etawa in East Java. One product is millet drinking. This is the pasteurization millet. Even in young and old people can drinking daily. The other product is millet lotion. Millet lotion and also solid milk bath and also liquid milk bath is minimized from chemicalia. But all of the ingredients in three kind of our product is most of them from natural resources.
My name is Wolfgang Knellen. I'm a professor of genetics at Kassel University in Germany. And I'm now here as a visiting professor funded by the DAD, the German Research Exchange Council. I've known this lab, Professor Patria's lab here at Brawijaya University for about 6 years, and I have always been very impressed how they are using high-tech methods and really state-of-the-art science to go into applications. That is to use the results from research directly for the benefit of people and to make products. By the society.
For Nutrilux body lotion, we use fresh goat milk. The goat's milk must be pasteurized, and then in different place, we prepare vegetable oil and butter. We heat the vegetable oil and butter in low until medium heat until they melt. And then we add stearic acid and glycerin to thicken the lotion. And then we mix this with the goat's milk solution. We add fragrance and olive oil. Blend one more time, about 1 minute, and then transfer the solution into the butter. We store the lotion in the fridge and use it within 2 weeks because we did not add any preservation.
This is a bar of Nutrilux, made from frozen milk and chopped into smaller pieces. Add a quarter of the lye and mix some more. Put your solution aside on the different place. Melt the vegetable oil and white butter, then add the olive oil to the melted oils. Add fragrance to the melted oil. Add the lye solution to the oil. Mixing the solution and finally pour into the mold.
We have 3 products. The first is liquid soap. The price is 15,000 rupiah. The second is lotion. The price is
Thank you for the moderator, Ms. Fatia Rahmatul Aziza. Good morning, everyone. Good morning, ladies and gentlemen. My name is Ana Safitri, and I will be your moderator for today's plenary session. Maybe you already known me from yesterday, so I'm here doing all-round job as a chair of the seminar, moderator, and also a presenter. For the first presenter for plenary session, we have Professor Fathia MKS PhD. Before she begins her presentation, I would like to read her CV. Please share screen the CV of Professor Fathia. Okay, this is CV for Professor Fathia PhD. So Professor Fathia PhD obtained her PhD degree from NIBB School of Life Sciences, the Graduate University for Advanced Studies, Okazaki, Japan. And then she became a lecturer in Brawijaya University since 1989 until today. She has a lot of impressive CVs here. Maybe this is a short CV from her. So I just read a few of her CV. So this is her job experiences. So from 28 till present, she's a head and founder of Research Center Monaginis UB, the research center that hosting ISMA 2020. And from 2012 until today, she is a director of Bioscience Institute from Brawijaya University. Go back. Slide. Okay, so she also published a lot of books, references on and textbook books. Here are some of the books that written by Professor Fathia. Next slide, please. And this is honors and awards for Professor Fathia as outstanding lecturer in Faculty of Science in Brawijaya University in 2014, and outstanding lecturer in Faculty of Science in Brawijaya University #1 in 2007. And then she also has a lot of publications with HI Index Corpus 8 and HI Index Google Scholar 11 and I10 Index 19, and a lot of published publication in high impact journals and also index in Scopus Index proceedings. Next one. Still has one more slide. Next slide. Third slide. You haven't seen the third slide. Third slide is also the publication. Okay, so without further ado, I think we should give Professor Fathia time to present her work. So for Professor Fathia, please, begin your presentation. Time is yours. Thank you.
Okay. Thank you for Dr. Ana Safitri to invite me and give me a chance to attend in this meeting. Assalamualaikum warahmatullahi wabarakatuh. Okay, today I would like to present, just a moment. Okay, this morning is second day of ISMAT meeting. I would like to presentation about the combination of nutrigenetic and nutrigenomic approach to deliver individual dietary. The concept of nutrition and genomic or genetic expression correlation is commonly, we know the common dietary chemical act direct and indirect to alter genes expression and structure. Some diet regulated the gene related with some disease. and also to play a role in onset and incident progression or severity of chronic disease. Dietary intervention according to the nutrition require and nutrition status and also genotype individually can be used to prevent or mitigation or cure the chronic disease. We can see in this figure our healthy control by our lifestyle and also nutrition and also environmental. Right now, we can explore and determine our genetic related to epigenome. For in this area, the environmental induce our genome expression. And we can also explore the transcription, proteomic, metabolomic, and also we know our phenotype. The definition of nutrigenomic is described the use of the functional genomic tool to probe a biological system following a nutrition stimulus that will permit an increased understanding how the nutrition molecule affect metabolic pathway and homeostatic control. In nutrigenomic science, we can focus on the effect of nutrient on the genome, proteome, and metabolome. We can see here, if we take a diet, some diet, maybe specific diet, this nutrient have a lot of chemical compounds inside. So this chemical compound can control directly to gene expression and also can induce the signal transduction inside cell. And the signal transduction connected with metabolism inside cell or tissue or our organ inside our body control indirectly to gene expression. When the abnormality in some disease, cell will grow abnormally. So this controlling from diet can control also the proliferation of normal cell growth. How is about the nutrigenetic? Definition of nutrigenetic is nutrigenetic study is to explore the difference of the genetic level influence of the response to diet. So in this different with nutrigenomic, firstly, we take the profiling of genetic our body or someone, and then, for example, in this figure, we can see in here about the single nucleotide polymerism. Even only single nucleotide is become different phenotype in our body. The simple we can see, for example, is anemia. Sickle cell anemia is just because only one of the single nucleotide polymerism changing, the structure of the blood become like different, not like what single cell, not circle anymore, and become structure it become changing. This for nutrigenetic, we explore a lot of genetic variant between individual result from numerous different in nucleate sequence within their genome in single nucleate polymerism. The most common form of genomic variation in human are more than 10 million SNP. And then why we learn about the nutrigenetic and nutrigenomic to take we have a personal nutrition. Nutrigenetic and nutrigenomic can be useful as tool to understanding of nutrient and gene interaction to develop of personal nutrition strategy to getting the optimal health and disease prevention. Now we not so familiar with personal nutrition in Indonesia, but I think in some country we have already taken the personal nutrition for some disease related with some disease. Personal nutrition is a diet addressed to an individual and based upon her on his genotype, nutritional requirement, and other factors, for example, ages and gender. It is expected that personal nutrition will prevent diet-related chronic disease. In this figure, in Indonesia, some people taking diet by keto diet, but some people, this group people for keto diet group is thinking we have a lot of eat meat but not vegetable. Actually, this Tamio is wrong because in keto diet we better still eat some vegetable. Like in this figure, we show you some the best vegetable for keto, but this other is not so good for keto diet. So how to achieve this goal? Basic study, firstly, we identify some biomarker. Actually, in my group, we have already started in 2008 to explore many gene profiling from Indonesian people, because as we knew, the Indonesian people have married with a lot of the other country, even in Caucasus, from Caucasus country, Africa and Chinese also. So we have a variable, genetic variable is very bright, different with other country. So we identify the difference of the SNP among this population. And then we got the expression profile. At the same time, we also explore a lot of the natural genetic resources from plant and also from animal. And then we take a study in functional nutrigenomic and also in nutrigenetics. We know right now we can learn a lot of omic science to understanding the relationship between nutrition and health, or nutrition and disease, or about nutrition induced to health and also induced to disease. In my group, we explore a lot of nutrient. That means nutrient in here is not only for our body, but also for our skins. That means in here is a cosmetic. So in my group, we explore a lot of bioactive compound of food, not only for our food and then we eat, but also we taking a lot of the nutrient compound to make the healthy cosmetic. Nutrigenomic, we can also in here explore the nutrigenetic, epigenomic, because induce environmental with the, and then we also explore the transcriptomic and other things. So nutrigenomic, we can see here, I think everybody have already love and like this food. This food, we know what is food? Yeah, that's right. This is hot meal with a lot of meat inside, I think. But remember, this is not so good food. Even some burger in here have vegetable. From nutrigenomic perspective, the bioactive food component, dietary signal that are detected by cellular sensor system. This time we focus in hypercholesterolemia and also obesitas. So we explore some genes like PPAR gamma and RXR receptor to control and then that influence the gene expression and protein synthesis and metabolic production. From this point, few gain dietary targets. The pattern of gene expression and protein synthesis and metabolic production in response to particular nutrient can be considered as a dietary signature. This is related with personal nutrition. So in my group also we explore metabolic syndrome, especially in nutrigenomic study. We know this is the metabolic syndrome. In here, some focus is obesity and also arthritis and also colon cancer, infection, diabetic, and other things. And we explore, like I mentioned before, bioactive compound to control in its tissue targets, even in neuron, and also DNA repairing, cell differentiation, and other. Now, one of these project in my group is to explore whole grain phytochemical from pigmented rice. We know this is a lot of pigmented rice. This project we joined with Professor James will present later after me and Saki. And also we join with Center or Institute of Padi in Indonesia, Indonesian Padi Institute in West Java. So we together explore because in Indonesia we have a lot variety in pigmented of rice. We can see some of them. This is already we explore black mentic. This is the different with even in Jawa, West Java, Central Java, and East Java. Black Toraja, this Black Toraja specific growing well in West Java. They have rich of anthocyanin and other component than other black rice. In Central Java, we know the jeliteng. Jeliteng also is a black rice. It's a good rice. No, they already, because the growing is not so well, they already plant culture to get the best standard of the plants. And we have a lot of red rice, red mentik, and red Aceh is specific also than other. And now ongoing research is red compo from Central Java and also from East Java. We already start to explore the content in there. And we have also the new variety, this purple rice and black mardas. Purple rice actually is from Korea, but black madras is actually from Japan. After the growing in Java island, they have a different profile with the original country. So we also already have both of them. We just start to explore the genomic, proteomic, and transcriptomic about them because it's a very, very different morphology with actually with the original country. So the red rice that as far as conclusion in until this time have a low glycemic index with high fiber and rich anthocyanin and a lot of vitamin and mineral, and also can blood glucose homeostasis. They have content like this. The black purple, the black or purple rice have a high fiber, rich anthocyanin, can cholesterol lowering, anti-obesitas, and anti-inflammatory. Interestingly, in purple, they have a 4.5 gram of fiber, a lot of the fiber than rice and also brown rice. Brown rice like this brown rice in Indonesia, have a low glycemic index and also can control the blood glucose homeostasis. This year, we try to publish our data. The MIT disease, MIT is metabolic syndrome disease, and bioactive compound that we explore from black rice to control the lipid process inside our body. So whole grain black rice, especially from wheat rice, the name is Toraja black rice, with have high content bioactive compound, regulated the PPAR gamma pathway, and reducing the visceral fat mass, controlling cholesterol and blood glucose, and also oxidative stress level, become a homeostasis and normal again. And then in this paper, I wrote the function of black rice from Toraja black rice is as antioxidant activity, anti-adipogenesis, and anti-obesity, and also can cholesterol attenuation. Nutrigenetic, we can show in here, genetic has a pivotal role in determining an individual risk of developing a certain disease. We can explore SNP very easy using PCR or RFLP and other methods. In complex disease, we focus in complex disease, in metabolic disease or degenerative disease. So we explore the SNP as the first time we start a long time ago. How to achieve this goal? Basically, now we know the GWAS, genome-wide association study. So when you see the video before I present, we explore a lot of the SNP, not only from human population in Indonesia, but also we explore a lot of genotyping from plant and also from animal, like cow and other animals. So for example, this is one I put in some publication, the role of personal nutrition in chronic degenerative disease. We can see here the people have, like me, a fat person with dysbiosis, and then she take a personal nutrition with Mediterranean diet. What is Mediterranean diet? Mediterranean diet is a focus on vegetable, fruit, herb, nut, bean, and whole grain. And the animal food is like seafood and other, but not so much for Mediterranean diet. And after some time, they take Mediterranean diet, this lady become thin and eusebiosis inside the stomachs. So Mediterranean diet have a function and anti-inflammatory, antioxidant activity also in here. So for nutrigenetic, we can see here, we can explore many profile of SNP inside the gene related with, for example, in here is lipid. And this, the profile of gene related with blood pressure and related with inflammation and also methylation. So what's the meaning of this? If related with cardiovascular disease, when the defect in gene related with lipid, so we take special nutrition or personal nutrition to control this lipid. But if this cardiovascular disease, the defect in blood pressure, so we control the personal diet related with blood pressure. And also in inflammation, methylation. This is also when we have obesity. Some people already eat, for example, in our research, getting the black rice can control the obesity. But if they have different profile of gene, so maybe the black rice cannot control for become a thin person, not to control the obesity. So we can examine or determine the profile of the person, what kind of profile of genetic in this person, and then to take the personal diet. For example, in here, for heart, we can analyze 13 of your gene, and that might play the important role. For example, this is some genes to manage our body overall for heart health. We can identify which one the defect of this disease. Each person sometimes is different with other person. For example, in here, we have a lot of data for bone health, controlling, and then we control with goat milk, especially in alpha-casein S2 protein in goat milk. So when they have a defect in bone disease, so this alpha-casein S2 can control some, for example, pathway in AGEs and receptor pathway, and this person is become normal again. So sorry, I will drink. I think in Indonesia, even insulin resistant taking in some disease in diabetic, but not so much in Indonesia for insulin resistant. So we can explore which one this gene profiling in this related with insulin resistant. We already have a profile in young diabetic. Young diabetic is our name, the juvenile diabetic in Malang, around 100 young people, and we control the expression of this defect of these genes, and know they can live better than before. The genetic assessment for antioxidant and detoxification. We can relate it with stress in here, for example, inosgenes, GST, and MSOD, and also SOD3. So we can control the food related with this cause defect of gene, and then analyze this gain when play important role in determine how your body manage related with antioxidant and detoxification of our health. And also we can control genetic related with inflammation, for example, EL, interleukin 6, and also TNF alpha. However, will the public health be improved with personal dietary recommendation? How costly will personal nutrition be? I have already discussed with the head of the healthy food in Surabaya. They can reduce the price of the personal nutrition. So this is one problem also for making the personal nutrition and then become a common diet in our population. Will the people be motivated to use a personal diet? At the moment, there is a degree of public confusion and resistance to message that foster unpopular advice. such as get more exercise and eat less calorie. Because some people are thinking in nutrient same meaning is calorie, but not meaning in ingredient. Important now to eat some nutrient is make the ingredient, what's the ingredient inside, so we become the healthy. Well, the approach to be available only for rich and well educated. How to educated people. So this is some proper to personal nutrition as a common dietary, healthy dietary. So short conclusion of my talk is the new omic science used in understanding the relationship among gen nutrition, health, and disease. Genomic focus on the effect of nutrient on the genome, proteome, and metabolome. Bioactive compound, for example, in my study in whole grain regulated the metabolic syndrome metabolism. Nutrigenetic determine the influence individual genetic markup in response to dietary component. Food with higher content of nutrition value and phytochemical compound has rich biological function and immunomodulatory factor to deliver healthy nutrition related with personal dietary. Thank you very much. This is ongoing research in my group. We have 3. This is with James and also this. And we also know to take stability and preclinical clinical testing to our cosmetic from alpha casein S2, protein, caprin, milk, yogurt, kefir, and healthy skin nutrition for anti-aging. Thank you very much. And then please Bu Ana, you can moderate this meeting.
Thank you, Professor Fatkia. That is an excellent talk, and I'm sure that we have a lot of questions for your talk because it is interesting about calories, obesity, food. We eat food every day. So we have already one question for you. The first one comes from Bu Tuti Setianingsih from Chemistry Department, Brawijaya University. So she asked about, this is her question, among all colorful rice or pigmented rice that you already mentioned in your presentation, which rice is the best to reduce fat? Is it easy to get this kind of rice in Malang? Thank you.
Okay, thank you, Bu Tuti, for your question. Yeah, I think in Malang we have is easy to even in market, a traditional market, and also in mini market and supermarket. So no problem for buy the black rice or red rice in Malang.
How about she asked about which kind of rice is the best to reduce fat or reduce obesity, maybe?
For obesity, we recommend we use black rice.
Black rice, okay.
Yes.
Good.
Then
So for
red rice, because I think in our population have thinking is black rice can also control the diabetic, but in my case, when I explore the bioactive compound from black rice in Java Island, is most of them could not control the blood glucose, but the success to control the lipid adipogenesis and also cholesterol synthesis and also obesity, especially in PPAR gamma gene.
Okay, yeah, okay. So we have another question from Ibu Cleo from Department of Gizi, Medicine Faculty, Nutrition Department. So she asked about, can you please explain in details how some vegetables are good or not good in your previous slides, maybe in the beginning of your slides, I saw it too, for keto diet based on nutrigenomics approach.
Yeah, in Indonesia that I mentioned before, some people when taking keto diet, they thinking better eat a lot of meat. But actually, the good nutrition is not only meat, but also we balancing with vegetable. So that I saw in my presentation before, they have some vegetable recommendation, but not for other, like in my presentation. So I will send to you also this paper for Leonara. Okay?
Okay, thank you.
Prof,
She has another questions. So because she is a dietitian, so as a dietitian, she wondered how to make a personal diet based on our genome become more easier.
That I mentioned in the last the last slide is a little bit difficult
Cheaper.
in Indonesia, yeah.
Yeah.
But I think next I will join with you and also Pak Bayu to making
App.
artificial intelligence to identify the genetic profile easily, so we can recommend to the personal someone, her or him, to take the personal nutrition. Okay, I think you already have the software for this, but only for identify the metabolic. Okay, so we can continue this project, and then not only for metabolic but also for genomic and transcriptomic. Okay.
Okay, so personal diet or personal dietitian doesn't really mean to be expensive. Now it's become more affordable.
That's right,
Yeah. Okay. I have a question for you. Is it all right? So because you talk about a lot of you already talk about a lot of pigmented rice like black rice, red rice, but I have question about what about white rice? White rice we always consume, not we always, we consume it almost every day. For white rice, so white rice doesn't have benefits at all, or it just make us full but doesn't have benefits at all for us to consume white rice. White rice.
Huh?
Sorry? Yeah.
Okay. Sorry. Okay. Thank you for your question, Anna Sang. Dr. Anna. Actually, the white rice have good benefit if the white paddy meal only one meal digiling. Because the brown rice in Indonesia different with other country. Brown rice is actually is white rice only one meal process. So they have a lot of fiber in there, and also can control homeostasis of blood glucose for white rice when no. But usually if eat the white rice with the white rice, this is the farmer getting 2 times or 3 times milk.
Okay.
So the brown layer is getting out because the brown layer in white rice is good for our healthy. Like this.
Oh, okay, yeah. Okay, so before we
Bu
finish the
Anna,
session,
may I ask to Prof. Fathia?
Okay. 2 more questions because we still have
one question. Bu Anna, is it okay?
Oh yeah, Professor Dian, monggo.
Professor Dian.
Prof. Fathia, congratulations, very nice talk, and I enjoyed with your presentation. Actually, I only agree with Cleonara and of course agree with you that personal diet is really promising, Prof, for the future therapy in the nutrition. But like Cleonara said that as a dietitian, sometimes we should make decision fast to do assessment based on our patient condition. So that's why maybe in the future it will be great if we can make like system to analyze quickly how the genetic type of from the patient. I think it's really wonderful idea if you could make like system to help a practitioner like us, dietitian, to identify the genetic type, so we can easier to find the personal diet. This just my comment, Prof. Fathia, from my perspective.
Thank you, Prof.
And
Dian.
then the second one, Prof, may I ask you and get your opinion? In your slide, you say that insulin resistant, for example, it will be good if we could analyze the gene like VDR, interleukin 6, the NF alpha, and PPAR gamma, I think, and ACE. You say that like one. And it should be gene analysis. My question is, sometimes we don't have enough money to analyze in gene level. What do you think if we only analyze this in plasma, Prof? Is it still representative to identify this one? Because I also read some paper also still use plasma for identification insulin resistant. Maybe your perspective to this problem. Thank you, Prof.
Okay, thank you, Prof. Dian, for your great question and also opinion. Yeah. For the first opinion that I mentioned, we will take a project together about the AI, artificial intelligence, because we have a lot already gene biomarker for this. So we can talk with Pak Bayu group and we can design of this. The second ones, I think for identify first by using plasma, it will be okay. It's a good way. But if you have, for example, I have a case in Prof. Joko group. They already reject some drug, but and then Prof. Joko sent me the blood from this patient, and then I identify the mutation of the gene related with diabetic. And also I take some analysis for among this gene and then the drugs. And the drug when enter to our this patient body is rejected, and the patient is become worse. So I talk with Prof. Joko group to take care them with different drugs. Because if the common drug for diabetic type 2 is this patient will be rejected for the effect of the drugs like this. So you can still using the plasma way to identify the insulin resistant, but if you have
like resistant or something, we continue to analyze deeper like that, Prof.
Continue to, this is the second way to another problems.
Yes, okay. Thank you, Prof. Thank you so much.
My
Thank you, Bu
pleasure.
Ana.
Yeah, thank you, Professor Dian.
Thank you, Bu Ana.
For the next question, I think I have to stop this discussion for Professor Fathia because the time is up. So for Professor Fathia, you still have actually 2 or 3 questions in the chat. So would you like
I will answer by chatting.
Would you mind to answer all the question in the chat? Thank you so much, because I have to call our next keynote speaker. So our next keynote speaker is Dr. Saki Shirako from Department of Biomedical Sciences, College of Life Sciences, Ritsumeikan University, Japan. Before Dr. Saki Shirako present her work, I will read her CV first. Sorry. Share screen. Okay, this is the CV for Dr. Saki Shirako, PhD from Ritsumeikan University, Japan. So she obtained her PhD from Kyoto University, Graduate School of Agriculture, Division of Applied Biosciences, in doctoral course in the field study of analytical chemistry, especially peptide and food science. For the master degree, she got from Kyoto University to Graduate School of Agriculture, Division of Applied Biosciences master course. So from the job experience, she's starting as an assistant professor in Department of Biomedical Sciences, College of Life Sciences, Ritsumeikan University from 2020 until now. So still a young researcher in the Professor Mikio Nishizawa group. So because here is from Professor Mikio Nishizawa groups. So she has some honors and awards from Young Investigator Award in ISO EFF in 2019, and then Bronze Award as oral presenters in 2018 in Global Food Science Student Competition in Jiangnan University, and also some poster awards. So the research of interest are analytical chemistry, food sciences, natural medicines, comprehensive analysis of herbal medicine from derived peptide. So I would like to ask Dr. Saki Shirako to start her presentation. And if you have help in sharing your screen, please let me know. Yes, can you try to test your screen? Yeah. Okay, we can see your screen now.
Can you see my slide?
Yes, I can see your slides.
Okay.
And I can hear you, but can you talk a little bit louder?
Okay.
Okay, now it's good.
Can you hear my voice?
Yeah, I can hear your voice.
Okay. And thank you for introduction. My name is Saki Shirako from Ritsumeikan University, Japan. I'm also dietitian, so thank you for giving a chance to make a presentation today. Today, my presentation topic is comprehensive analysis of crude drugs of Japanese Kanpo medicines. First, let me introduce about Kanpo medicines. Have you ever heard of Kanpo or Kanpo medicines?
Not really. In here, we call it herbal drugs.
Kanpo medicines are commercially available as Kanpo extract products as shown in this picture like this. There are many types of Kanpo medicines in Japan. For example, these red 4 products are for stomach problem such as stomachache or heavy stomach. The other 5 products, these green ones, are for stress. So as you can see, there are many types of Kanpo medicines and Kanpo extract products in Japan. Today, my presentation topics are as follows. First, let me introduce about Japanese Kanpo drugs and crude drugs. Kanpo medicines is traditional Japanese medicine which have been used up to date. And each Kanpo medicine consists of several crude drugs. Most of them are herbal drugs. And Kanpo formula is the recipe for the combination of Kanpo crude drugs based on Kanpo medicine. The crude drugs are derived from medical plants, herbs, animals, and minerals. Many Japanese medical doctors prescribe Kanpo medicines. Now, Kanpo medicines are commercially available in Japan. We can obtain them not only by prescription from medical doctor, but also in pharmacy by ourselves. They are transmitted from ancient China to Japan in the 50th to 60th century. And they have been modified, improved, and developed to adapt to Japanese people. The formula showing adverse effects have been eliminated. Now, 184 formula are available as Kampo extract products in Japan, like this. The recipes and quality of Kampo formula are strictly defined by Japanese pharmacopoeia. Each crude drugs includes a major constituent and principal constituent. The major constituent is included at the high content and characteristic of a crude drug. On the other hand, principal constituent shows pharmacological activities. As I said, the principal constituent shows pharmacological activities. However, most principal constituents are still incompletely identified and characterized. So we are thinking that they are sources of new smart molecules and sources of new drugs. Okay, for next, I will introduce our research on pharmacologically active constituent from Kampo medicines. We have established a method for separation and identification of pharmacologically active constituent from Kampo medicines. There are 3 steps: extraction, separation, and identification. Here I show the scheme for determination of the chemical structure of bioactive constituent from Kampo medicines. First step is extraction step, as I marked in red. Medical plant used for Kampo crude drug, are extracted with absolute methanol under reflux, as shown in this picture. The methanol extract were evaporated under vacuum, as shown in the right pictures. Through these extraction steps, we can obtain methanol extract. In most cases, they are powder form like this. And then methanol extract are fractionated by crude fractionation, which is called ABC fractionation, using ethyl acetate and butanol and water. In ethyl acetate superfraction, which is A fraction, hydrophobic constituent are included. And in N-butanol superfraction, which is B fraction, amphipathic constituents such as glycoside, saponins are included. And in water superfraction, which is C fraction, hydrophilic constituent such as sugars, amino acids are included. Then the next step is separation and identification step. After extraction and ABC fractionation, we screen their pharmacological activities based on nitric oxide assay, like shown in this picture. By screening their activity through nitric oxide assay, then the active fraction can be further separated by column chromatography to obtain pure chemical compound. The structure of this compound can be analyzed by mass spectrometry, NMR, and so on. We can also perform animal experiments to test the pharmacological activity of pure chemical compound or these extract or fractions to determine their bioactivity in vivo model. Now, let me introduce about nitric oxide assay in our lab. In our lab, anti-inflammatory effects of extract or fractions or compounds of medical plants are screened based on nitric oxide suppressing effect. Nitric oxide is produced from arginine, one of amino acid, by inducible nitric oxide synthase, INOS. In our body, lipopolysaccharide LPS, inflammatory cytokines can also stimulate the expression of INOS gene to produce nitric oxide. However, excess production of nitric oxide causes liver disorders such as inflammation, infection, septic shock, and skin allergy, and so on. In our laboratory, ex vivo model of liver injury is used for nitric oxide assessment. Briefly, primary cultured rat hepatocytes are collected by collagenase perfusion of rat liver. Treatment with pro-inflammatory cytokine, interleukin-1 beta, IL-1 beta, stimulate the production of nitric oxide in primary cultured hepatocyte. Then the medium cell extracts and total RNA can be collected. So we can check whether treatment with or without constituent of campo drugs, extract, or fractions can inhibit or decrease the nitric oxide production, expression of INOS gene, and cytokines, and so on. Can you hear my voice? Okay.
Oh, yes, I can hear you.
Okay.
Yes, continue.
From here, I introduced an example of nitric oxide suppressing compound found in ABC fractions. Many constituents, such as oils, terpenoids, flavonoids, can be found in this ethyl acetate solvent fraction, hydrophobic fraction, and shown to have high pharmacological activity. One example of compounds are from Palila leaves. Have you ever tried sushi in Japan or Indonesia?
Yes.
Thank you. Palila leaves in Japanese shiso or aojiso. It's sometimes used as a garnish with sushi, as shown in this picture. They are components of several campo formula that are effective for patients suffering from allergic disease. Through the extraction separation processes, our previous study identified that schizoflavonon A from Palila leaves, and we found that they have nitric oxide suppressing activity. As you can see in the right figure, treatment with IL-1 beta only extensively increased the production of nitric oxide. However, treatment with cisoflavonol A suppressed the production of nitric oxide in a dose-dependent manner. iNOS messenger RNA was also induced by IL-1 beta only. However, cisoflavonol A decreased IL-1 beta induced iNOS messenger RNA level. Furthermore, cisoflavonol A decreased IL-1 beta induced messenger RNA expression of TNF-alpha, which is one of inflammatory cytokines. Other 6 compounds were identified from ethyl acetate sorbol fraction, which is A fraction of Pallilar leaves, and performed nitric oxide assay and calculated IC50 value. So for now, I introduced our research on identification of bioactive constituent from compo drugs based on nitric oxide assay screening. Except for these Pallilar leaves, many constituents from hydrophobic fraction, A fraction, have been identified and shown to exert high pharmacological activity. In contrast, few studies have focused on hydrophilic fraction, water-soluble fraction, which includes hydrophilic constituents such as sugars, peptides, and amino acids. In the final section, I would like to introduce especially my new project focusing on peptide. For a short break of my talk, please imagine the digestion and absorption rate of what you eat, especially protein. These 2 pictures are, as many of you know, Indonesian dishes.
Yes, it's Indonesian dishes.
Satay and nasi goreng. And the other picture shows Japanese traditional diet, rice, miso soup, and some other dishes. Many of you might imagine that protein in foods are degraded into peptide, and peptide also degraded into amino acid for digestion and absorption. Yes, it is true, but there are new facts in our field. This figure shows the absorption rate of food-derived protein. As you know, proteins are digested into amino acid or D and tripeptides by protease or peptidase. As a result, about 80% of D and tripeptides are transported into intestinal tissue through peptide transporter, so-called peptide-1. In the tissue, these peptides be digested or degraded in amino acid in peptidase in the tissue. Totally, most proteins are finally absorbed into blood circulation system as amino acid form. However, still approximately 10% of G and tripeptides are resistant to this kind of peptidase digestion and absorbed directly into blood as peptide form. The peptide, which is resistant to peptidase, have been found to have biological functions in addition to being sources of amino acid after food ingestion. These peptides are called bioactive peptide, and recently more and more researchers, including me, are interested in this peptide. So what is bioactive peptide? One example is hydroxyproline containing peptide from collagen peptide. This figure shows collagen peptide supplement in Japan. And the right table shows the list of structure of food-derived collagen peptide in human blood after ingestion of this kind of supplement. It means that these kind of hydroxyproline containing peptide are absorbed into our blood circulation system directly as peptide form after ingestion of this supplement. Today, let me introduce about pyroglutamyl peptide, which I have studied from my student days. The amide in glutamine is easily released from carbonyl group and coupled to converted to pyroglutamic acid like this. The amino terminal glutaminyl residue of the peptide is also converted to pyroglutamyl residue, which we called pyroglutamyl peptide. Because their N-terminal is blocked and not like normal type of peptide, this kind of pyroglutamyl peptide can survive protease digestion. This graph shows the content of pyroglutamyl peptide, especially pyroglutamyl leucine, in many fermented foods in the world. Very interestingly, pyroglutamyl peptides are highly distributed in Japanese fermented foods, such as Japanese rice wine, soy sauce, and soy-based miso. Soy-based miso is used for miso soup cooking as shown in this picture. Many effects of the administration of these kind of pyroglutamyl peptides have been found in vivo and also in vitro experiment, such as antidepressant-like effect, hepatoprotective effect, anti-inflammation, attenuation of DSS-induced colitis, and increase of host antimicrobial peptide in rats. Just briefly, I will introduce one biological function of pyroglutamyl peptide. This is a summary of my research in my student days. I found that oral administration of pyroglutamyl leucine to rats fed on high-fat diet normalized the disturbance of gut microbiota in colon. As a mechanism, what I found was that pyroglutamyl leucine was absorbed into small intestine, especially ileum, the last part of small intestine, to stimulate the production of antimicrobial peptide. Antimicrobial peptide is low molecule protein that help modulate gut microbiota. And I concluded that administration of pyroglutamyl peptide at this very low doses modulate gut microbiota by increasing host antimicrobial peptide like this. So this is one of the effects of pyroglutamyl peptides. So how we can know and how we can identify bioactive peptide? In our body, peptidases in end proteinase digest could be further degraded by exopeptidases in pancreatic juice, lumen, epithelium tissue, and blood during digestion and absorption processes. As shown in upper figure, these are list of peptides found in corn gluten hydrolysate. And as you can see, most peptides are not affected by end proteinase digestion by pepsin or trypsin. However, very surprisingly, by digestion with exopeptidases, most of them are degraded. However, some peptides like pyroglutamyl peptide, PEQ or PEP, or other peptides still can exist. The same peptides can be released by in vitro digestion using carboxypeptidase A and using aminopeptidases. Peptides in the in vitro digest of exopeptidases, including above these peptidases, could be candidate for the peptidase resist peptide in body. This left figure shows the exopeptidase resist peptide found in wheat gluten hydrolysate, WGH, in vitro. And as you can see in the right figure, same peptide such as pyroglutamylproline, PEP, and polylglycine, PG, could be detected after ingestion of wheat gluten hydrolysate in human plasma. So summary of our pre-identification method of peptidase resist peptide are like this. In vitro digestion by end proteinase, such as pepsin, corresponded to digestion in stomach or small intestine in our body. Following in vitro digestion by exopeptidase, such as using aminopeptidase, corresponded to peptides which are absorbed into blood or our whole body. So we are thinking that exopeptidase resist peptide in vitro experiment could be a candidate for bioactive peptide after ingestion of food or something like that in our body. There are studies on food-derived peptide. However, there are few studies investigating the presence of peptide in compo drugs. And also, there are few studies considering digestion and absorption processes of peptide in compo drugs. So we are starting a new project on identification of exopeptidase resist peptide, which could be a candidate for bioactive peptide in compo drugs. This is a method. So hydrophilic fraction of compo drugs were used. Following end proteinase and exopeptidase digestion in vitro, end proteinase resist peptide and also exopeptidase resist peptide were collected. And also non-digest compo crude drug were prepared. By using LC-MS analysis using precursor ion scan mode and other scan mode, we detected the presence of peptide by using this machine. Today, the time is limited, so I just show our preliminary result. This is an example of pre-identification of exopeptidase resist peptide. Black line shows non-digest, just crude compound drugs. And blue line shows end proteinase resist peptide. And the red one indicates exopeptidase resist peptide. Please look at the peaks labeled yellow in here. As shown in this figure, you can see some peptide existed in endo, the blue line, but this appeared after exopeptidase digestion. So this figure suggests that these peptides in blue one were degraded or digested by exopeptidase. Next, look at the peak marked in yellow in here. As shown in this figure, you can see this peak still could be detected after exopeptidase digestion. So it suggests that non-digest black one and end proteinase blue one still contain exopeptidase resist peptide. And next, look at this peak around retention time 15. And very surprisingly, as shown in this figure, you can see new peak appeared after exopeptidase digestion. So it suggests that exopeptidase resist peptides were generated after during digestion and absorption processes of crude compound drugs. So far till now, I checked 7 compound drugs from compound drug A to G. This figure is just one example of comparison of exopeptidase resist peptide of 7 compound drugs. Very interestingly, there are differences in distribution of exopeptidase resist peptide in compound drug. They are peptides that all compound drugs have in common. However, they are distinct peptides also exist in compound drug. Our preliminary findings are as follows. We found that the presence of exopeptidase resist peptide, including pyroglutamyl peptide in compound drugs. And there are differences in distribution of exopeptidase resist peptide between compound drugs. This is a summary and conclusion of my talk. We are interested in Japanese compound medicines. By nitric oxide suppressing assay, we have been identified new compound from compound medicines. Animal experiment related to inflammation, such as septic shock, were also performed and confirmed. As a new target, we are starting to identify peptide as a new candidate for smart molecules in compound drugs. Here, acknowledgement, Dr. Mikio Nishizawa is professor in our lab. He gave me an opportunity to make a presentation today. And Dr. Kenji Sato from Kyoto University, he is my previous supervisor in Kyoto University. I got interested in bioactive peptide under his instruction in my student life. Thank you for your attention. Terima kasih. Thank you.
Thank you, Saki-san. That's a very good presentation and a lot of combination work in analytical chemistry, organic chemistry, biochemistry, and medicinal chemistry about Kenpo drugs, Japanese drugs. So I would like to open question to all of the participants here, but in the chat there is no question so far, but maybe if you have questions, you can ask Dr. Saki directly, or I will start asking question about your work. Can I start question first, Saki-san? Is that all right? Okay.
Yes.
So because you present a lot of work in analyzing Kenpo drugs medicine. Oh, we have questions. So maybe I will read question from Dr. Tomohiko Sasase from Kyoto University, from your alumni university. So he asked about, this is his question. I just read his question. So after identifying new exopeptidase resist peptide, how do you evaluate the pharmacological effect of the peptide? How do you predict the type of efficacy, and how do you know the anti-inflammatory or anti-diabetic or anti-hypertension? So what are the biological activities of your compounds?
So thank you for question, Sasase Sensei. And in the cases of Kenpo medicines, we already know the effect of Kenpo medicines. Can you hear my voice?
Yeah, I can hear you. It's very clear.
So I repeat again. So
Yeah.
in the case of Kenpo medicines, we already know the effect of Kenpo drugs. So if we identify new exopeptidase resist peptide, we will synthesize the peptide or to synthesize the peptide to the company, and doing animal experiment regarded with the effect of crude Kenpo drugs such as nephrosis or anti-skin allergy and so on.
Okay, thank you very much. So you already know the effect, a type of effect of the peptide, and so you retest the pharmacological effect of the isolated peptide.
Yes.
Okay, thank you very much.
Thank you.
Thank you. So we have next question for Professor James from SUT Thailand. So he asked, how much do the resistant peptides contribute to the pharmacology of the Kenpo drugs compared to the non-polar components?
Thank you for the questions, Dr. James. But unfortunately, this work was just started from this April, so I just estimate the structure of exopeptidase resist peptide. So from now, I want to determine the content of exopeptidase resist peptide. So now I cannot answer to your question about the content. I'm sorry.
Oh, yeah. I think you still have continued questions. So Professor James, if you want to ask yourself, or I read your questions. It's all right if you want to ask yourself, or
okay, maybe I can talk.
Yeah.
Can you hear me?
Sure.
Okay.
Yes, I can hear you very clear.
For dipeptide, 400 is not that many to synthesize, right? So I would think that some company or some lab somewhere has already synthesized all 400. So can you get that type of a library to test in your assay, say your nitric oxide assay or whatever assay you want to test?
Yes.
I mean, is it commercially available or academically available, a library of 400 peptides?
Yes, some peptides, especially the peptide, can synthesize by company or by ourselves, so we can get easily. But in especially pyroglutamyl peptide, There are few reports, there are reports, but not so much report about their effect. So if you want to check the bioactive activity of pyroglutamyl peptide, we often synthesize by ourselves.
Because I would think that any peptide that you synthesize with glutamine on the N-terminus will convert itself to a pyroglutamyl peptide just by automatic reaction.
By heat treatment with low temperature and long reaction, the glutamine in N-terminal converts to pyroglutamyl peptide.
Okay, thank you.
Okay, we have one last question. So this is very last question before we go to the next speaker. So from Cleonara from Brawijaya University. So we've already know that fiber contain foods such as fruit, vegetables, nuts, and etc. But would they affect the microbiome by its probiotic or prebiotic roles? May I ask you about how peptides could change the microbiota? Is that the same pathway of bioactive peptides, like probiotic benefit for microbiota? I hope you understand the questions.
Okay.
I don't understand.
Thank you for the question. And yes, there are some compounds like prebiotics or probiotics that modulate our gut microbiota. However, as I shown in my slide, the effective dose of pyroglutamyl leucine is just 1 milligram per kilogram body weight. This ratio is very, very small compared with prebiotics or other probiotics. So in cases of prebiotics, they will be digested by microorganisms to generate some effective compound in our stomach. However, we thought that in the cases of pyroglutamyl peptides, the dose was very low, such as 1 milligram per kilogram. So we think that this peptide stimulates some cell to produce some good compound for our gut microbiota. And as I shown in my slide, we found that pyroglutamyl peptide at a very low dose stimulate the cell in ileum, the last part of small intestine, to produce active compound named antimicrobial peptide to modulate our gut microbiota. Other previous study suggests that the decrease of, in the disturbance of gut microbiota, the decrease of antimicrobiota was founded. So we are thinking that pyroglutamyl peptide stimulate the production of antimicrobial peptide to help modulate the disturbance of gut microbiota. Is it answer for your question?
Yes, I think it's maybe, yeah, I think it's because she's also dietitian like you. So yeah, that's why she asked about microbiota or gut microbiota. Okay, thank you, Dr. Saki Sirako for your presentation and all your answers. So we need to move to our last speaker for plenary session. So for the last speaker for plenary session, we will have Professor James Ketudat-Keynes, PhD from Suranare University of Technology, Thailand. So before Professor James begin his presentation, I would like to read his CV. So here I have his CV. Let me select doing slideshow. Okay, so Professor Keynes obtained his PhD from University of California, San Diego, United States. And then he also did his postdoctoral fellow in the same university and then he became a professor in School of Chemistry, Institute of Science, Suranare University of Technology, Nakhon Ratchasima in Thailand. So here is his job experiences. So recently there's a lot of job experiences here. So right now he's appointed as head of chemistry department in Suranare University Technology, and then he also visited University of Brawijaya in 2017 as a visiting professor in the grant of award professorial, if I'm not mistaken. And then he has a lot of publication. Here are just some of his publications. So in Scopus, with its index of 22, here are some of the publication in highly reputable journals. For example, in 2020, in Protein Expression and Purification Journal. And you can see here, if you can see, there's a lot of publication for Professor James. And Professor James, Professor Vatskia, and myself we have joint supervision too. We have students together here. So Professor James and our group has a very strong cooperation, especially in the field of study about rice, pigmented rice. So Professor James, are you ready to present your work? So please welcome Professor James.
Okay, thank you very much, Bu Ana, and also to Professor Vatskia for inviting me to talk in this session. And so I'm a little bit shy because I feel like we had two very nice talks so far today, and I feel like mine's a little bit disorganized comparatively, so I hope you'll get something out of it, or at least since most of you are online, you know, you can check your email or whatever if it gets too boring. So let me share, if I can get my share up here. Share screen. Okay, and so my talk is about carbohydrate active enzymes from rice and related compound discovery and application. This may be a little bit misleading because actually I'm not going to go into that much of the, I'll go into a little bit of the scientific application of some of the compounds that we've made from using our enzymes basically. So I always start with a slide about my research interest, and basically in our lab in Suranare University of Technology, we mostly worked on carbohydrate active enzymes, and for the past 20 some years it's primarily been on rice, but lately we've been going more in looking at human and bacterial beta-glucosidase and also some other, say, biomass degrading enzymes from bacteria. At the same time, I help out with Chulaporn Research Institute. Professor Jisnusen Sawati is the head of the lab there in looking at metabolic disease in Thailand, and also helping some other geneticists around Thailand for evaluating the effect of their mutations in their patients on the protein that's actually causing the activity. So in our group, we went everywhere from, mostly we would collaborate to see anything from the knockout rice to producing the protein and going to the enzymology and determining the protein structure. Okay, so I'll go through sort of a little bit of that in my first half introductory session, and then I'll get into some parts about making transglycosidase products later. So this all began about 25 years ago when I met up with Professor Jisnusen Sawati at Mahidol and Chulaporn Research Institute. And at that time, they were looking at extracts of Thai plants for glycosidase activity. And the idea was they were going to make oligosaccharides, such as prebiotic oligosaccharides, maybe some glycoside for cosmetic or for non-ionic detergents and that type of thing. And they started out screening in the bottom here, Thai plants, especially seeds, are easy to come by for various glycosidase that they could find. So I was joining about that time. And again, the basic idea is they look at these enzymes that can do either hydrolysis or transglycosylation. So they could go here, maybe this is water coming in and some aglycone, some alcohol coming out from the glycoside, or it may be a sugar. And basically the reaction is reversible, and we can also sometimes get something other than water coming in here to transglycosylate. And then they look at various sugar specificities, and eventually we look at the rest of the molecule that's being hydrolyzed, the aglycone part. So when I joined, we started chatting, and we said, okay, they were working on this Thai rosewood, or Dalbergia cochinchinensis beta-glucosidase, beta-fucosidase enzyme. So I was basically a protein chemist that had some molecular biology experience. So I said, okay, I could help to clone that. And Professor Jisnusen had a student named Morat Champatanachai, and Dr. Jantragan Srithomsap were also working on this project. So we helped each other to clone, and eventually we came out with this paper in JBiochem in 2000. And then after that, my group followed up, and we sort of took a different approach. Instead of just looking for the activity, we looked for the genes. So we were look at the conserved parts of the proteins in the families, and then we make degenerate primers and start amplifying genes from these different plants. And we, for instance, worked on another Dalbergia species and found another enzyme that was interesting in that it was a disaccharidase. I mean, it had beta-glucosidase activity, but it preferred to cut the diapiosyl glucoside instead, so disaccharide. And eventually, we did another further work on that as well. So that sort of followed up. So along that time, I had a student come into my lab by sort of chance. Was a very good student, gold medal student at Konkan University to the north of us. And she said, I want to work on rice enzymes. And I said, well, I work on beta-glucosidase. You could look at some of those from rice. Naively, when I began, I thought, oh, all the people in Japan and Taiwan and China have characterized every possible feature of rice by now. But it turned out that, in fact, there was no complete beta-glucosidase sequence from rice at the time. So we kind of finished that, and we expressed the protein in E. coli, and were able to characterize it with this paper here. So I'm just showing you some of the papers that began quite early on in the lab, okay, and in the effort that we're talking about. And I should have another paper on here. Eventually, we did look at some transglycosylation features of that enzyme as well, such as transglycosylating vitamins and such, but we didn't really go into the further application from that. But while we were working on that, we found the rice genome project, Arabidopsis genome project were going on, and we could find that we had some advantage that we could get some cDNA sequence, and we didn't have to use the primers from degenerate primers anymore. We could use the specific sequence, which made the cloning a little bit easier. And then we found that the whole genome was being completed, and we identified about 40 sequences in the genome, but a few of those were endophytes, a few of them were pseudogenes, so about 35 genes in one family, glycoside hydrolase family 1 in rice. And we could categorize them based on either their relationship to Arabidopsis sequence. We made about 8 clusters of genes that are found both in Arabidopsis and rice, or by enzymes that had been characterized. So eventually we characterized about 10 or 15 of those, these shown in green mainly. Also one from barley that had been characterized but not cloned previously. So they had different features. Some of them were not actually beta-glucosidase, like hydroxyisourate hydrolase actually cuts a bond in a purine. and some of them it turned out to be transglycosidase. So I'll talk about that more later. So typically, when we are doing this work, we would clone out the gene and we put it into expression vector like PET32, which has thyroidoxin histidine tag for purification, and then some protease site, and initially enterokinase. So our first beta-glucosidase we first called bGlu1, but eventually we gave a more systematic name, OS3bGlu7, based on the position in the genome, chromosome 3, seventh position from the first chromosome going through. Anyway, so we were able to finally find this system in origami DE3 E. coli that would produce the protein. It was a little bit of a struggle because secreted plant protein is not the easiest thing to produce, but eventually we could get it produced. So this is another example, not bGlu1. This is what we call OS3bGlu6, and this is just showing the typical method of purification that we would grow the cells and induce the protein and extract the protein and then purify by immobilized metal affinity column based on the his tag. And in this case, we had a tobacco etch virus protease site instead of the enterokinase site. And then we could cut, basically we run it over the IMAC column and get this big band, but a lot of junk. We cut with the TEV and then run it back over the column, and most of the junk gets removed by the column and our protein came through. So that was quite effective for us, so that we could go on to do enzymology and crystallography from that method. And this is just showing you, typically we would screen both, I didn't show the slide, with some glycosides, synthetic glycosides, but maybe some oligosaccharides as well, to see what's the best substrate for these enzymes. And eventually we solve structure. These are the first four structures we solve in this particular family. Okay, and I don't need to go into the details of that today. So the idea for application or making product, again, is we have the enzyme and the substrate coming together, and then they form what we might call E star, the covalent intermediate, where the glucose is left on the enzyme, but the aglycone already left. And then we have the second substrate come in, either water for hydrolysis or some other, usually alcohol or other nucleophile for releasing in transglycosylation. So we could call that P hydrolysis, P transglycosylation. So we could manipulate this system in order to prevent this part of the hydrolysis. One way that we did was to mutate the catalytic acid base. And then if we have a good leaving group, the first step to form the covalent intermediate can go on, but we need a better nucleophile than water for the second step. So we could just produce a glycoside based on that, because water's pretty good nucleophile, but not that good, okay? This is just the wild type mechanism again. Another way we had for making oligosaccharides in particular is to mutate the nucleophile residue. So now there's no nucleophile, but we still have acid base. And then we put alpha link fluoride onto our sugar, and that would cause the, look like the covalent intermediate basically. And then we just get one step to transfer to some other oligosaccharide or whatever. So this is called glycosynthase. And we did this type of thing, but most of our enzymes made something like cello oligosaccharides. So this is just couple control and low substrate concentration curve. And this show you what happened if we had a high amount of donor substrate. We get these long oligosaccharide that precipitate out of solution. This show you that we separate by chromatography. We could see the various peaks. And if we use the mass spec, we could get out to about 11 glucose residue in a beta-1,4 link chain from this enzyme, which was our original bGlu1 with the catalytic nucleophile mutant. So this allows us to do the transglycosylation, but it's a little bit of work, and we have to use sort of unusual substrate, okay, unnatural substrate. So the other thing we could do with these type of mutants is the acid-base mutant. We could put the oligosaccharide into the active site and kind of figure out what the how the enzyme interacts with the substrate, and that was quite useful for us. And we could also do various synthesis with that as well. And we could do that. We did a lot of analysis of the active site binding, both for glycosynthase and normal hydrolysis reactions. But again, this is good for learning about the interaction, but it's not necessarily an easy way to produce some useful oligosaccharide or glycoside. So what we went on is to look at the transglycosidase specific enzyme, which turned out to be one OS9-BGLU31, we called it. And basically this enzyme was my student Jiep Suganya Luang was working on, and it was a very terrible beta-glucosidase. We got very little release from just about any substrate. But I say, okay, do the pH optimum. And we noticed that the acetate buffer gave much higher activity than something like citrate or MES. And this was quite similar to what happened in our mutant enzyme, say acid-base mutant, where we would get rescue with acetate was typically a reasonable rescue agent. So I said, hey, maybe this is causing transglycosylation. So Jiep took the products from various points and ran the TLC. And in fact, she could see this transglycosylation product, which is the glucosyl acetate, okay, beta glucosyl acetate. And so we saw that it's really transglucosidase. It doesn't like to hydrolyze so much, but it likes to do transglycosylation. And we were pretty proud of ourselves for about a week maybe. And then it turned out that I saw in Plant Cell this paper coming out from Professor Ozeki's group in Tokyo. And basically, they had just found that their glycosylation of the second sugar onto anthocyanin, okay, so going from anthocyanin, which has 3-glucose on it, anthocyanidin 3-glucoside, would then come from an acyl glucose using an enzyme in the same family, okay? And then it's, depending on which plant, it could go on the 5 position or the 7 position. So, okay, that was a little disappointing. We weren't the first, but at least it gave us some ideas about what kind of substrates to test. So we went on to look at that, and we could test a bunch of these phenolic acid esters that they had used, and they were nice enough to provide us with some of them. We see that they were actually quite good donors of glucose and also great acceptors of the glucose, we'll show later, okay? And even something like some flavonoid like fluorizin or apigenin 7-O beta-glucoside, or even phytohormone GA4 beta-glucose ester that we had in the lab could also act as donor for the glucose to some extent, as well as the PNP glucose, the synthetic substrate we were using originally. So we went on and characterized that enzyme in the plant in terms of my collaborator, Johnson John in Korea, and Thai student working with him found some insert lines, and we could show that when it's knocked out, we knocked out the gene. This is just showing you these lines don't have any production of this RNA. And then we could see that when we do LCMS, we got extra peaks coming up. And in fact, one of them is the 1-ferrol beta-D glucose. So what we knew is a good donor substrate. And then later we saw that if we look at fatty acids, not that much effect, but if we look at fatty acid glucose ester, when we knock out the gene, they get much higher because they're basically almost undetectable in the natural rice. But when we make the mutant, we could detect extra amounts. So we don't know what those are doing in the rice, but at least we can see that somehow our enzyme is modulating the amount, okay? So that was just the sort of biological effect that we were interested in. But we also wanted, because we were biochemists, we were thinking that we want to know the difference between hydrolase and transglycosidase. So we look at, we made a homology model based on BGLU6, one of the structures we had that had glucose covalently bound, actually 2-fluoryl glucose to keep it on. And then we look at some of the residues close to that where the water must bind to release the glucose. And we could see that, okay, some of them were more hydrophobic And this tryptophan was a little bit further out, but we thought, okay, it's a very important position for the substrate specificity of some of our other enzymes. So we went ahead to mutate that as well. And we just changed them to see if we could make it a hydrolase. In fact, we're pretty much unsuccessful at that. But what we did find was that, in fact, this W243N mutant was much more active than the wild type. Okay, so this was basically changing it to look like our BGLU1 hydrolase. And it didn't make it a hydrolase, but it made it more active in terms of overall activity. And then if we normalize to look at the using ferulic acid as an acceptor, and we use all these different compounds, we could see that, yeah, depending on the mutation we made, we could see a certain difference in specificity. Our active mutant tended to like some things more than wild type, and some of the other mutants tend to like things less. Okay, so we went on to examine that more, and basically what we could see was not only did we get more activity towards something like ferulate, but in the wild type, we basically get one product, which is the glucose ester product here. But if we had the W243N mutant, we could get a glycoside. So basically, I didn't draw the picture here, but basically it would be putting the glucose on the other end of the molecule over here. Okay, not as much as the glucose ester, but still it shows sort of a more broad specificity for this enzyme. Okay, so we could then imagine that we could synthesize more compounds with that. So we went on to examine that more, okay, looking at various acceptors again. And what we did was we made more mutations of the W243N position, and none of them, the initial set was as good as the original W243N, but we could see, we also look at like when we use a flavonoid like camphorol, there are 4 different possible acceptor sites, so do we get a difference? And indeed, the wild type is only making basically 1 or 2 and a small amount, but if we use a mutant, we could get more. Actually, one of them comes out in the same position as the substrate, so you can't really see. We could see by mass spec though. And then we get some bis-glucosides, 2 different positions of glucose on there. Okay. So basically, we went on to mutate every possible amino acid at that position. And then we identify a few more that are quite active. And the other one that was quite active is W243L. Okay, and W243L is similar to W243N, but on some substrates, it's more active. Okay, and it turned out that both of those are also quite stable enzymes, even compared to the wild type. And so we had tried to crystallize the wild type, but we didn't quite get it. Okay, this is just showing you some more mutants that we made. Not really need to go into those details. But anyway, I'm getting ahead of myself. So we did look at application. One of the thing is we thought, okay, some antibiotics, maybe they'll be improved if we put glucose onto them, or they'd be more soluble because some of them are a little bit limited by solubility. So we did screen some various antibiotic, and we could see that chloramphenicol could be glycosylated to produce the chloramphenicol glucoside, and we isolated it and showed by NMR that it's the glucose on the primary alcohol position, which is not too surprising, given that that's easy to access. And we also were able to make, so the only problem with this is it's nice and soluble. It actually was patented in Japan in about the 1960s to chemically do this, but the problem is it completely inactivates the antibiotic, so it's not that useful unless you can find a beta, because chloramphenicol binds to the ribosome, so it's probably not getting through the membrane into the bacteria. So unless you can find a bacteria that wants to hydrolyze it for you, it's probably not so effective. But anyway, it showed in principle that we could do this. Tsunari was helping some high school students on this project. And we also could do some phytohormone like ABA, which was of interest to our lab. So this product, maybe we're not going to get much money off of this, but okay, this is what I was saying earlier. We found that when we did the homology model and we did docking, we didn't get very good result in terms of the position. So we eventually were able to solve the structure for the W243L mutant. And this is just comparing it to the BGU6, OS3 BGU6 hydrolase enzyme again. And so now we're looking at docking into that structure to try to see if we can engineer it for maybe more broad specificity to maybe glycosylate some beta-lactam antibiotics or other compounds of interest, okay, some anti-cancer drug, for instance. So the other objective we had is to try to get it to use a more cheap donor like Cellobios, because we have some complaint. We can able to publish that previous work in Green Chemistry, but which is quite high impact, but some reviewer will complain that, well, it's not very green using PNP glucoside. You should be using something either more atom efficient or natural. So we thought we better find some better donor substrate for our enzyme. So basically, we're still working on that. We haven't got very far on that, but at least we're set up to do it. However, what we found was we could make use of the product of our enzyme for our own research. So normally, if we want to buy abscisic acid glucose ester, ABAGE, it costs us about $1,000 for 5 milligrams. But we could not do very much work with that. So we could synthesize that with the PNP that we could get glucoside that we got cheaply from China, and our ABA can get fairly cheaply. And we could make the glucose ester, and we put a patent in on that for just to show that we could do it. And so we used that to screen various enzymes from rice in our lab, and we could see that the ones that had the highest, a lot of them have activity on this substrate, it turned out. The one that had the highest were these two OS4BGU12 and OS4BGU13, which had previously been identified as phytohormone beta-glucosidase, but not with ABA, with other phytohormone glucoconjugates. So we looked at that, and we had expressed those. In E. coli, we could do the kinetics and everything to show that it's reasonable. But some other enzymes are quite closely related in this bunch here. We call 9, 10, 11. So they're all, if you look at, this is a chromosome map at the bottom, they're all genes right next to each other on chromosome 4 of rice. So maybe they're doing something similar or basically redundant function. So we couldn't produce the other ones in E. coli or yeast. So we put them all into Arabidopsis. And then when we put them in Arabidopsis, we could see that if we extract the Arabidopsis and look at ABA glucose ester, they all have higher than the wild type plant for hydrolysis activity. And also we had gibberellin GA4 glucose ester in the lab, so we test that as well, and also higher. And with a synthetic substrate, a bit higher, but maybe not quite as much higher as with the phytohormone glycoconjugates. So we think that they have at least potential to act on these type of compounds in the plant, okay? And in fact, we can see a little bit of these look very sick Arabidopsis, but basically these are grown with ABA. If we grew them with salt media, it would be similar. That the wild type looks kind of small, but the one that has overexpression of the beta-glucosidase is a bit bigger, and this is sort of typical response. Again, it sort of depends on the treatment, but in this case, for these two, we could see bigger plants compared to the normal plant. And this is obviously a synthetic system. It's not real, but it does show that they can maybe mediate some stress response in the plant. Okay, so now the last few minutes, I want to transfer over to a different use of these products of our BGU 31. Okay, OS9B2. So basically, we started a project with rice phenolic compounds, including some black rice compounds with Professor Faccia. And the work that had been done in Professor Ozeki's lab on anthocyanins was also showing another action of these glucose esters, okay? So basically, if you have the anthocyanidin that's synthesized in the cytoplasm and maybe has 1 or 2 sugars put onto the 3 position, and then gets transferred into the vacuole of the plant cell. And then this p-hydroxy benzoyl glucose, like this, acts as a donor for another glucose on the 7 position in delphinium. It's a nice blue flower. And then after that, the same substrate dominates the other half. It donates this p-hydroxybenzyl group onto the glucose that was just added. And then another time is going to donate the glucose again, and then another benzyl group, etc., until we get a very extended anthocyanin called cyanodelphin. Okay, and Professor Ozeki's group characterized the enzyme that looks like our GH1 hydrolase, like similar to a bGlu31, and also the acyl transferase that's transferring the acyl group, which is called SCPL type acyl transferase. So we got interested in those, and we can make the substrates easily with our enzyme. So we started to look at those. Again, this work is not from us, this is from Professor Ozeki's group again. I forgot that I had to click that again. Okay, was wondering where that sign went. Okay, anyway, that's their work. And other groups have worked on these type of enzyme, and they have a lot of different glucose esters that can be donating to different kinds of compounds to make, not just anthocyanins, but lots of other compounds can get acylated, or saponin or tannin or whatever. So basically, it's making sort of more complex compounds from different organisms. Again, where it's characterized, it's found in the vacuole. So we saw there's not much been done on that in rice. So we tried to identify some from rice. So if we look at the family, half of the family is the serine carboxypeptidase enzymes, and this part down here is the serine carboxypeptidase-like acyl transferase group. And again, some of the monocots from oat for making saponin have been characterized, but the ones from rice have not. So we just basically, I don't really need this other phylogenetic tree, but it just shows some other, which ones have been characterized so far. So we looked at the expression of some of the rice enzyme, and we found these 2 that we thought might be interesting. One we called OSSCPL2 and one we call OSSPL7, because they were found in the flower bud that could have some color, but OSSPL7 also in the young grain. So maybe it's likely to have anthocyanin on the pericarp of that grain in the so basically we thought these might be interesting, although this is from white rice. So basically they could be modifying other compounds, flavonoids, or who knows, other cell wall or whatever, we don't know. So eventually we expressed those in E. coli, not very successfully. So ISTI was able to finally express the OSSPL7 in Pichia. And this is still very preliminary, but I just wanted to show you that we had another application for our synthesis, where we can again take something like p-hydroxybenzoate and make the glucose ester with the bGlu31 enzyme, and then we can analyze it and go this slide from ISTI, so a little more animation than I usually bothered. Anyway, so we can see that we also made several other possible donors for it, okay? And I just show you the first one that we ran this through UPLC, and we're using the cyanidin 3-glucoside anthocyanin as an acceptor. Is not necessarily the natural acceptor, because this is an acceptor in some other plants, but not necessarily in rice. So we could see at least that the the acceptor substrate somehow had 2 peaks in what we bought from the company at this wavelength, but at least they're going down and we get some small peaks coming up. So we still need to do some more work because these may just be degradation products, but we can see that it's specific for the reaction that has enzymes. So we're hoping that we're getting some acylation of the compound, but we need to screen a lot of other substrates as well. Okay, so that's the end. Sorry, it's a bit preliminary on that part, but again, I just wanted to see that sometimes you can use the enzymatic activity that you discover in the lab to make more reagents to work on further projects. And I think that's probably useful for a lot of people to do. So just to conclude, the glycosidases, or glycoside hydrolase we call, can be used to produce oligosaccharides and glycoside by transglycosylation. And discovery and engineering of the rice OS9-VGU31 transglucosidase allowed us to enzymatically synthesize a variety of glucose sides, glycosides, glucose ester and glycoside. And we have used the cheap production of the phytohormone glucose conjugate to investigate the enzymes responsible for hydrolysis or maybe transglycosylation of those compounds in rice. And phenolic ester production also allowed us to investigate these serine carboxypeptidase like acyltransferase. Okay, so basically what we want to do is eventually really characterize them, and then maybe we can apply and engineer them to modify various compounds for increasing bioactivity or bioavailability, and also, basically either by glycosylation or acylation. Sometime by removing the glucose, we can as well. So that's basically all I want to say. I'm 10 seconds over, but I should at least thank you for listening and thank Professor Fatjia and Buana again, and Professor Jisnussen for starting and mentoring all this. Professor Dr. Marina, a lot of researchers, and my students and postdocs, all shown. Current group in this picture, I mentioned some of the others along the way, various collaborators and some funding source. So thank you very much. I'm about 38 seconds over, so thank you.
Thank you very much, James, for your presentation. A lot of work in your presentation about the enzymes. So I would like to invite to all participants who has questions to Professor James. But if there is not yet questions, maybe I will start to ask question first to you, James. Okay, so about the enzyme itself that you talked to us about beta-glucosidases from rice, because you took the enzyme isolated from rice. Is it specific rice, like white rice indigenous from Thailand, or pigmented rice that Professor Fatjia talked about?
Yeah, so that would be interesting, but right now is basically we're getting all these as genes from the genome. So basically most of the genome work is done on the the Japanese rice, right? So basically Nippon Bare, famous genetic model for rice, right? So basically this is a very good point, because I would like to get more information on the black rice. In fact, several Indonesian, Thai, other Korean, Japanese black rice have had their sequence, genome sequence, but it's all just available as crude data in the BGI IRRI collaboration. They did 3,000 plus cultivars of rice, but they didn't annotate, they didn't assemble the genome, they just put the raw data available. And I'm not enough of a genome bioinformaticist to get that information. Actually, I wanted to get that information out and I haven't. Probably someone in the world should have done that by now, but I couldn't find it yet. So basically, it would be very interesting to look, because something like, I didn't mention, but Dewi, the collaborator with you and Professor Fatjia, Dewi had looked at one glucosyltransferase that we thought might be involved in anthocyanin synthesis, right? And
Yes.
in the white rice, it has an intron, but we don't know if it's a real intron because most glucosyltransferase genes don't have an intron. So I was curious if we look in the black rice, maybe it's not there, and maybe that's means it's more active, right? Or maybe it's really active and the white rice is not. So yeah, it would be nice to look at some of the others. But sometimes the cloning is not as easy as we hope. And sometimes we did amplify another glucosyltransferase from black rice recently, but we haven't sequenced it to see whether it has any significant difference from the white rice gene. Because we just use the sequence from white rice for amplification. Okay. So anyway, yeah, everything's basically white rice right now. Some of it we did amplify from Thai indica rice, so it's slightly different, but normally it's only maybe 1 or 2 amino acids different anyway compared to the Nippon Bare sequence.
Okay, thank you. So we have now question from Pak Eko, but it's quite a lot of questions. So I read it and you can also read by yourself because it's quite long. I just worried if I misread the question. So he asked, glycosylation is the most important mechanism transferring sugar moieties from sugar donors catalyzed by ubiquitous GTs. As far as I know, he said, uridine diphosphate glycosyltransferases, GTs, can use activated UDP sugar donors such as UDP glucose, UDP xylose, UDP arabinose, UDP galactose, and etc. So how about lipophilic acceptor molecules? Can we use sterol UDP glycosyltransferase to modify the sterol compounds? So I think this is a question from student to his supervisor.
Yeah, ECMO is another Brawijaya associate that's working with me right now. Right?
Yes.
So actually it's true. A lot of these things, because different plants do things in different ways, like for the anthocyanins, and some of them use the UDP sugar-dependent glucosyltransferase, right? Which is quite common. The reason we didn't use it as much is because we think the substrate is more expensive, basically. So if we can get glycosidase or glycosidase-related enzyme to do the transglycosylation, we can use cheaper substrate to provide the sugar, right? But in fact, it's true. We can use the UDP glucose-dependent glucosyltransferase to do it. And Day we did. We put the gene into expression vector, and then she was able to produce the glucosyltransferase. Just that, you know, depend on my budget. I don't know how much UDP glucose or UDP xylose or whatever I want to buy, right? So it's a little bit expensive compared to the typical glycosidase substrate. Yeah. So, but yeah, it's true. It's the natural way that most things get made. But we have this other way that happened. So basically that happens mostly in the cytoplasm, a little bit in the Golgi. And then we have this other mechanism in the vacuole, which is more water soluble and more, well, we can produce the substrates from relatively cheap compounds in the lab. So that's why we're trying to take advantage of that.
Okay. Okay, so for Pak Eko, if you still have more questions to Professor James, you can ask him by yourself, I think, with like intense discussion in your lab. Okay, so thank you very much once again for Professor James Ketudatkins from SUT. So I also thank to all of our keynote speakers today, Professor Fathia, Dr. Saki Sirako, and Professor James. So I will handle time to our moderator. She will guide us for the next session. Thank you, moderator.
Too bad.
Thank you so much. Ladies and gentlemen, what an excellent presentation from our keynote speakers. And now we invite you to join the session with the links are being described on your screen. But before we go to the oral or parallel session, now we will hold on a photo session. Two keynote speakers as well as participants who are welcome to take a picture. And I will count from 3 to 1. 3, 2, 1. All right, thank you. Okay, very good. 3, 2, 1. All right, thank you. Now we invite you to join the parallel session. And before we go to the parallel session, let me remind you to attend the closing ceremony after the parallel session to enter the Zoom link, because we will announce some awards before the closing ceremony. That's all from me. Thank you.
Bisa. Oh iya. Dah. Oke, sih. Kan udah selesai lah. Oh, nah ini. Sir, I want to go in the charge. Aku nak ingin dulu, tak ingin. Tak ingin dulu. Aku kan sok belok chattingan ni.
الحمد لله الذي أنزل على عبده الكتاب ولم يجعل له عوجا قيما لينذر بأسا شديدا من لدنه ويبشر المؤمنين
Welcome once again to the second International Seminar on Smart Molecule of Natural Resources, or ISMART 2020. It's very nice to see all of you once again, and we hope you have been enjoying yourself at the seminar so far. We are very honored to have with us here this afternoon an Excellency, distinguished guests, participants, ladies and gentlemen. We have had a productive and inspiring time together, and this event about to come to the very end. I hope you found the presentation on this conference are informative and helpful. Now, before we go to the closing ceremony, we will announce awards for the best presentation. The best presentation award of ISMART 2020 goes to. We have 5 winners for this best presentation. The first best presenter goes to Farah. Based analytic devices or micropad methods for determining hydroquinone in facial lightening cream using chlorogluconol reagent. The second best presenter goes to Ernawati Sinaga with the title Hepatoprotective Effect of Rhodomycosis tomentosa fruit juice in breastfed with high-fat, high-cholesterol diet. The appeal or Percy americana meal extract in rats with dyslipidemia. The fourth best presenter goes
They have learned many new aspects of research in the related topic, either from keynote or general participants. In addition, collaborations between Universitas Brawijaya and other universities, both from Indonesia and overseas, can also be initiated during these 2 days. This year, we had participants registered to this event from not only Indonesia, but also from overseas. From Indonesia, we have delegates from Universitas Brawijaya, Universitas Airlangga, Universitas 11 Maret, Universitas Hasanuddin, Universitas Syekh Kuala, Poltekkes Medan, UIN Sunan Gunung Jati. From overseas, we have delegates from Kyoto University and Niigata University. I would like to also express my sincerest gratitude to our keynote speakers. Our keynote speakers were Professor James Ketutat Chance from SUT Thailand, Prof. Fahrul Zaman Huyop from UTM Malaysia, Dr. Saki Sirako from Ritsumeikan University Japan, Prof. Dian Handayani from UP Indonesia, and Prof. Fathia from UP Indonesia. It is worth noting that ISMAT 2020 event generally highlighted the important topics in the field of natural genetic resources. Importantly, to address important issues nowadays, that is outbreak of the coronavirus. The ISMAT 2020 could not become a reality without the help and assistance of many parties. Thus, in this occasion, I would like to sincerely thank the Rector of Universitas Brawijaya, all members of organizing committee who have provided meaningful help and assistance for the implementation of this conference. And last but not least, I would like to thank all the conference participants that contributed to making this the most memorable ISMAT 2028. Finally, I hope that you can sign up again next year